Zinc, very important trace mineral. The functions. So zinc is having it is a component of more than 200 metalloenzymes. The important metalloenzymes are carbonic anhydrase, carboxypeptidases A and B, alkaline phosphatase, alcohol dehydrogenase, glutamic dehydrogenase, lactic dehydrogenase. And zinc is an essential for many nucleic acid synthesis. For example, DNA polymerases, RNA polymerases. And for the synthesis of these nucleic acids, zinc is important. It is playing an important role. And for the proliferation of the lymphoid cells, zinc plays an important role. The role of zinc on immunity. It plays an important role in bactericidal activity. It regulates the phagocytosis by macrophages and neutrophils. As we know, the macrophages and neutrophils are the important factors which plays a role in immunity. And zinc also plays a role in regulation of cytokines and antibody productions. And the zinc also has a role in the biological activity of interleukins. And zinc is necessary for the activity of some of the immunity mediators like thymolin. This thymolin promotes the T lymphocyte maturation. And zinc is a major intracellular regulator of lymphocyte apoptosis. That is the natural killer cell activity. And zinc is a part of many proteins. There are 1,000 known proteins associated with zinc. And this, as I already told, the zinc protein is involved in transcription and translation of genetic material. And the deficiency of zinc in rapidly growing tissues will reduce the DNA, RNA, protein synthesis, and it will impair the cellular division, growth, and repair. Zinc is having a role in metabolism of fatty acids, carbohydrates, and nucleic acids. And it is a part of testosterone, synthesis of this insulin, prostaglandin metabolism. Zinc is having very vital role in skin, health of the skin, because it is involved in nucleic acids, skin nucleic acids, and collagen synthesis. In relation with vitamin A, zinc is involved in the normal functioning of the ovary. For the rupture of the follicle and the oocyte maturation, for ovulation, zinc is controlling these functions. Hence, in collaboration with vitamin A, it is actually controlling these functions: ovulation, oocyte maturation, and follicle rupture. And it is also having a control on the normal vision. For the conversion of the retinol to retinal, zinc is required. You can see this diagram. So it acts as a cofactor along with the dehydrogenase to convert the retinol to retinol. So if you can just imagine, if the zinc is absent, so these are all the functions which are going to be hampered. It is also involved in the reproduction. For the normal spermatogenesis and secondary sex organ development, zinc plays a role. In females, adequate concentration of zinc in the serum and in the diets are vital for uterine involution and tissue repair after the parturition. And there are some research I would like to share. In Australia, the sheep grazing on zinc deficient pasture supplemented with 140 milligram of zinc weekly increased the lamb production in relation to the ewes which are not treated with zinc. And in 1919, Minson has done some work. In goats, the consumption of low zinc diets has led to low conception rate, prolificacy, and zinc supplementation has increased the prolificacy by 14%. Minson in 1990 has done some work in rams that were fed ration with 2.4 ppm of zinc, which is actually a deficiency, the low level. 2.4 ppm is low level, which has resulted in atrophy of the seminiferous tubules and complete inhibition of spermatogenesis was observed. So zinc plays a key role in embryo implantation also. And zinc is having role in neurogenesis, neuronal migrations, synaptogenesis. Zinc is known for destruction of cancerous and precancerous cells. It is involved in healing of the wound. And we use the wood powder, which is having the zinc. Whenever we are hurt and if there is a bleeding or wound, we use the zinc powder because it is having the role in healing. And it is zinc is also plays an important role in membrane stability of the RBC. So one of the research work that was conducted in Bangalore in our college in 2017 by Ramchandraya et al. He has supplemented the zinc to growing glands. They have made 3 groups, T1 which is control, T2 and T3 group, wherein T1 was getting the standard diet, which is based on finger millet straw and compounded feed mixture. That is a standard diet. It is a control group. T2 was a group which was supplemented with 100 ppm of zinc. T3 was a group which has given 200 ppm of zinc. We can see the average daily gain. The group T2, which was receiving 100 ppm of zinc extra, was showing 103.2 average daily gain compared to the control group, which was significantly different from the control group. Whereas in the T3, which was receiving 200 ppm of zinc, has shown 102. So there was no significant difference between T2 or T3. So that means they have concluded that additional supplementation of zinc at the rate of 100 ppm will give 100 grams per day. That means 3 kgs of extra body weight gain can be achieved. And in 2019, Suresh et al., he was my student, under my supervision, we have conducted a trial of zinc supplementation in goats. In control group, there was no supplementation, only the ICR recommended zinc level, that is 40 ppm. But in T1, we supplemented zinc at the rate of 100 ppm. In T2 group, we have supplemented the 2% urea, and T3, we have supplemented both zinc as well as urea. We have got the satisfactory result in only T1 group, that is 90 grams per day was achieved, the average daily gain, which was significantly different from control. So overall, I would like to say that additional supplementation of 100 milligram or 100 ppm of zinc has a good result. So coming to the deficiency of zinc, zinc deficiency leads to hyperkeratosis, parakeratosis. You can see in the pictures, rough hair coat, alopecia, and emaciation. The animals will be very weak. There will be decrease in the, even if the intake is good, dry matter intake, daily feed intake is good, but they suffer from emaciation. You can see the inflammation of nose and mucosal hemorrhages, nostrils, thickening and cracking of the skin around the nostrils. You can see here the skin eczema in and around the eyes, the alopecia you can see. So this is a very common symptom of zinc deficiency. The dry scaly skin on ears is also seen. And you can differentially diagnose this with the parasitic infestation. So even during mange, the same type of symptoms are seen. You can scrape it out and go for parasitic diagnosis, or otherwise it will be usually the zinc deficiency leads to this type of lesions. The cracks in the skin of coronary bands around the hoofs is one of the symptoms. So stiffness of the joint, the swelling of fetlock, these are the other symptoms. Development of the horny overgrowths of mucosa of the lips is seen when there is a deficiency. And even the Ibrahim et al. In 2016, he has conducted a work, the effect of zinc deficiency on the blood parameters. The WBC count was significantly decreased from 0 day to 20th day with the decrease in zinc. And you can see the lymphocyte count, lymphocyte percentage was also decreased with zinc deficient status. The sources of zinc, these are all the sources of zinc. The yeast is a source. Legumes are rich in zinc. Germs of the cereal grains are rich in zinc. Brans, wheat bran, rice bran, these are all rich in zinc. Corns are rich in zinc. Colostrum is rich in zinc. The bioavailability of the zinc from plant source is relatively poor due to their phytic acid content. The phytic acid content will not allow the zinc to get absorbed. So the bioavailability is poor. But the animal byproducts such as meat meal, fish meal, they are usually a richer source of zinc than plant source. The commercial sources of zinc are zinc sulfate, zinc oxide, zinc chloride, and zinc carbonate. Among them, usually we use zinc sulfate as or zinc oxide. And the bioavailability is good in zinc oxide, but the solubility is less.